I'm Vaughan.
And I'm Brendan.
And we are actually going to present one of the general interest papers in functional proteomics. This particular paper is about how proteomics has been applied to the field of molecular paleontology.
So if you are not familiar with what molecular paleontology is, it is actually the study of ancient biological remains at the molecular scale. So that is, we are going to be looking for small molecules like DNA, carbohydrates, lipids, and of course proteins. And these are going to be found in fossils via primary or secondary methods of detection. So in this field, this would be undertaken, for example, to provide evidence to support the notion of evolution or adaptation between extinct or extant species. In 2007, John Asara and Mary Schweitzer and their research group published a paper in the prestigious journal Science. In this paper, they report that they had isolated a protein via a primary method of detection from fossilized remains using proteomic methodology. In addition, the results that they had obtained suggested that there may be an important evolutionary link between the dinosaur Tyrannosaurus rex and the modern-day chicken. So this was the first time something like this was published and reported in the literature. So as you can imagine, it generated a significant amount of interest in both the scientific and public communities. The name of this paper was Protein Sequences from Mastodon and Tyrannosaurus rex, revealed by mass spectrometry. As I previously mentioned earlier, the process used to acquire their data was via proteomic methodology. The first step, of course, is to acquire the proteins out of a fossilized sample. And the second step is then to analyze them on some sort of database that has the potential to describe evolutionary links. Schweitzer herself is a paleontologist, and she had access to the bone samples, which were an ostrich femur, which acts as a proof of concept, a 130 to 600,000 year old mastodon skull, and the femur from a 68 million year old Tyrannosaurus rex. So in bones, the major protein constituent is collagen, and this actually forms greater than 90% of the bone's protein mass. In addition to being readily available in bone, collagen is actually quite hardy. It has a significant biological half-life and is a highly conserved sequence, which actually make it an ideal candidate for a molecular paleontological study. So to extract peptides from bones is actually not too dissimilar to a standard extraction protocol. You can see the flow chart on this slide. You will take your bones and grind them down to a powder, remove the calcium, extract the proteins, digest them to peptides, and then purify and concentrate those peptides, and then deliver them via a microcapillary LC system to a mass spec where you would perform top 10 analysis. Normally, you would actually match these spectra against a database using your algorithm of choice. However, as you might have already surmised, the genomic sequences of dinosaurs are not readily available. In addition, any potential evolutionary drift in the intervening tens of millions of years may not even work even for highly conserved sequences like collagen. So in order to get around these hindrances, Asara and his group developed what he describes as a two-step proteomics process. And this is actually best explained using their own proof of concept species, the ostrich, which at the time did not exist in any genomic or proteomic database. First, they took their spectra and they utilized Sequest to search against the NCBI non-redundant database. They received 87 tryptic peptide matches, which are shown on the screen, related to extant species. And this mainly shows collagen alpha-1 type 1 from chickens. So the overall coverage of collagen alpha-1 type 1 is highest at 33%. So it becomes designated the protein of interest. The second step is to generate a theoretical sequence of peptides that may approximate change that may have occurred through evolution. And this is done by taking the top three matching organisms for alpha-1 type 1 collagen and then building predicted sequences from them. So for ostrich collagen, alpha-1 type 1, this is the chicken, the frog, and the newt. And they weighted one organism based on the initial search parameter. And in this case, for ostrich, it was the chicken. Then you will apply the predicted sequence algorithm, which works the following way. So if all three residues are the same, you keep that residue. If two of the three are in a majority, use that one. And if all three are different, use the weighted one. And you can actually see an example of this on the slide. In addition to the above, they also applied a point accepted mutation matrix. And this was applied to residues that could not meet consensus. So in those cases, they selected the weighted species residue and they used the PAM 100 matrix to generate a residue that was acceptable via evolutionary change. So now that you have a predicted spectra that has been generated, you can actually rerun the earlier analyzed spectra against this new sequences and receive additional results. So in this case, the ostrich peptides identified an additional 6 sequences, 4 that matched the consensus weighted chicken sequence and 2 from the point mutation consensus weighted chicken sequences. And to validate these sequences, they were synthesized and refragmented. And you can actually see on this slide the side-by-side comparison of the synthetic to the actual isolated peptides. So these additional sequences bring the total collagen coverage for ostrich up to 39%. And according to Asara, this reveals enough to ascertain difference between an ostrich and a chicken, and then also confirms that this provides a mean to assess divergent evolution between species.
From the mastodon sample, the researchers identified 74 tryptic peptide MSMS spectra that matched to collagen sequences from mammalian organisms in protein databases. When combined, these spectra provided approximately 32% coverage for the collagen A1T1 peptide sequence and 29% coverage for the collagen A2T1 peptide sequence. An additional 4 peptide sequences unique to mastodon were also identified. 2 of these were identified using the organism-weighted consensus method, and 2 were identified using the point-assigned mutation-weighted consensus method. The 4 sequences were then confirmed by comparing each of their experimental spectra to the spectra of a synthetic version of the peptide. The very good alignment of the major ions between spectra confirmed that the spectra were real. With the addition of the 4 unique peptide sequences, the amino acid coverage for the A1T1 collagen sequence was increased by approximately 5%, from 32% to 37%. From the T-Rex bone extract, 7 peptide MSMS spectra were identified that could be aligned with the predicted fragmentation patterns of collagen A1T1, A2T1, or A1T2 sequences from various extant vertebrate taxa in protein databases. A blast search alignment and similarity search was performed on the 5 A1T1 collagen peptides as a group against all taxa in protein databases. This search found that the group of T-Rex A1T1 collagen peptide fragments showed a 58% sequence similarity to chicken, 51% for frog, and 51% for newt. As with the mastodon sample, the researchers successfully used spectra from synthetic versions of the peptides of interest to confirm each experimental spectra. The conclusions made in this paper are, first, the abundance of sequenceable collagen in the approximately 500,000-year-old mastodon sample may indicate that the sequenceable protein can last much longer than the previously thought limit of 1 million years. Second, from the T-Rex results, the researchers proposed that the closest living relatives to the T-Rex based on the species with collagen sequences available in the database used at the time is chickens. It should be noted that at the time, the databases did not contain the collagen sequences for other closely related taxa, such as alligators and crocodiles. The third conclusion was that they had demonstrated the method for obtaining theoretical peptide sequences for taxa that have not had their genome sequenced. Since being published in 2007, the paper has been cited 131 times. Further, the number of citations has been relatively consistent over the years. Within the first 16 months of being published, there were 3 separate rebuttals to the paper, with 2 of these being published in Science, the journal where the paper was originally published. These rebuttals and many other unpublished works, including blogs and news articles, put forward arguments that the results, namely those from the T-Rex sample, may not be valid. To quote Carl Sagan, extraordinary claims require extraordinary evidence, and in this paper, evidence such as this was not provided. The first rebuttal, published in Science, was authored by a group of 27 mass spectrometrists, bioinformatics experts, and dinosaur experts. This rebuttal criticized the T-Rex protein analysis in the paper. The criticisms they raised can be seen in the table, namely, doubt about the collagen surviving due to the expected half-life of collagen in the environment where the fossil was found, the lack of accounting for the risk of contamination from collagens of other species, a lack of providing proof of sequence authenticity, which came in the form of incorrect data interpretation, and not enough data being included. However, they did find that the mastodon sample and its results were sufficiently investigated and valid. In the second rebuttal published in Science, Hesner, a computational biologist, criticized the lack of proper statistical testing of the T-Rex results in the paper and that a full list of the spectra was not made available. To quote him, "Indeed, statistical significance, e.g. false positive rate, is a number that needs to be computed, but Asara et al. Never described how they computed statistical significance, and it's not clear whether they tried." So, should the paper have been published? In light of the many criticisms alleged against the paper, we believe that the paper should not have been published in the state it was released in. Instead, it should have been put under a more stringent review so that the issues raised could be identified and addressed before it was published.